Participants, we have to wait for 6 minutes. Sir will join soon. Good afternoon, Dr. Surjit.
Good afternoon, Surjit sir. Okay. Am I audible?
Yes, sir. Yes, sir. You are
Okay.
audible.
Okay, fine. So I'll start from my this thing, my presentation right away.
Great. Uh,
So do you want some intro kind of
yes, yes, yes.
Yes, yes,
Great,
sir.
great, sir.
Yes, wait, sir.
section and all?
Yes, yes, yes. So warm welcome all
Okay, please go
participants
ahead.
in AICTE Training and Learning Academy sponsored 1 week faculty development program on drug engineering. So eminent speaker for session number 6 is Dr. Surjit Debnath. I'd like to give introduction. Dr. Debnath is currently working as a head Department of Medical Laboratory Technology, Women's Polytechnic, Tripura, with more than 13 years of experience in biomedical research and high-level teaching. Dr. Debnath participates in various academic works in institutions of Germany, Denmark, and Hong Kong, besides in premier India's labs such as AIMS, IIT Kharagpur, IIT Guwahati, NBPGR. Dr. Debnath is the recipient of Young Scientist Award from different agencies like DBT, DST, CSIR. Recipient of Tata Trust Grant for CME course in the Royal College of Pathologists, London. Reviewer of several Scopus and SCI Index journals published by Elsewhere, Springer, Biomedical Central, along with the regular academic activities. He is also engaged with several biotechnological companies in northern and western India as a consultant. He have conducted various workshops and talks, acted as a session chair in an international conference sponsored by national and international academic agencies. He delivered numerous invited talks in national and international academic gatherings. So I'd like to again warm welcome to our eminent speaker for this session. Welcome, Dr. Sujit Debnath, sir. And I would like to hand over this session. Dr. Sujit, sir.
Okay, so I'll be putting this thing.
Yes.
Okay. Yes. Is it
Yes, yes, sir. It's visible.
okay? So when we are talking about this thing, peptide-based drugs, we are actually talking about protein engineering for desired therapeutic potentials. Okay. So what has happened is my lab is working on this thing, protein engineering for various purposes. One of the domain is peptide based drugs. Okay, so these are the major aspects of peptide based drugs, which are either used for diagnostics and then therapeutics and then eventually drug delivery mechanism as well. In diagnostics, more mainly we are talking about bioimaging and biomarkers, whereas in therapeutics there are many, for example, like regenerative medicine, cancer therapy, antibacterial agent, antimicrobial agents, and so on. What has been a new focus in this particular trend where diagnostics and therapeutics are being given hand in hand in a single, and we are talking about theranostics, where in a single engineered protein there are ability to bring a different kind of usage, diagnosis as well as therapeutics. When we are talking about only diagnosis, then we are essentially talking about proteins which are having fluorescent properties and which are useful for various kinds of fluorescent based diagnostic usage. And then for biosensors, in biosensor we are essentially talking about peptides and proteins and enzymes which can have a specific sensing ability due to the inherent capacity of the enzymes. For example, some redox enzymes. Given one example, cytochrome oxidase, which is in plenty in use in all kinds of bioelectrodes that we use. For example, like detection of glucose in blood, and several other things that is usually today mainly uses cytochrome oxidase because it is a redox enzyme and can bring about rapid changes in the oxidation reduction potential, and therefore it is having plenty of usage. Then that of if we are talking about therapeutics, then regenerative medicine, wound healing, it's a very good application. Let me give you one specific example for the pharmacy people. If you go to a shop and ask for a medicine, that is this thing, Placentex. Placentex is used mainly for the treatment of what we call is bed sore. In geriatric patients, what happens is they usually have pressure ulcers because for a long period of time, the geriatric patient will be lied on bed, and therefore some pressure ulcers that are developed on the skin. In order to get rid of this, Placentex is being used. Now, what is Placentex? Placentex is nothing but combination of various growth peptides or growth hormones, growth factors, which are available in the placenta. Okay, now placenta, as you all know, is the sac where the embryo develops. The embryo develops from a single cell, and then several other stages are there. Eventually, it becomes a complete animal, isn't it? So in that case, what happens is several growth factors are needed. Now, in order to repair the degraded skin due to the pressure ulcer, you need to also use factors. But if you purify the growth factors, the cost will be very, very high. So the company, what it did was it collected all the embryos and the placenta and then made into a gel. So what has happened is they have increased the stability of the growth factor. The growth factors are also very less stable. So you need to do something so that the stability of the whole peptide increases. So what they did is they embedded the this thing, what you call is placenta extract into a hydrogel, and therefore it made into a complete what they call is solution, which will give you a very good repair and regeneration, but also in a very minimum cost. Okay, so accordingly, drug delivery methods are also many. If I give you specific example, like liposome mediated drug delivery, or they are peptide mediated drug delivery, are there, which we will talk in specific discussions. Okay, so these are some major aspects. There are several other aspects as well. So here we are having some very good examples of engineered antibody. For example, immunotoxins and immunocytokines and all. For example, if I give you a specific example of rituximab, any of you would have definitely have the idea of it. Now, rituximab is a monoclonal antibody which is targeted towards SLE, systemic lupus erythematosus, an autoimmune disease. But today, as you know, that rituximab has also been used for treatment of kids, that means young age patients who are suffering from nephrotic syndrome and then glomerulonephritis kind of things. Okay. Then there are side examples like for breast cancer today, there is an excellent therapy which is called Herceptin. Now, Herceptin is again an immunotoxin, an engineered antibody in which there is a diphtheria toxin which is embedded on it, and it acts like a magic bullet because it identifies only specific antigen which is developed in the breast cancer and no other cell, and side effects of what we call is chemotherapy can be reduced. In chemotherapy, what happens, as you know, that it is not very much targeted. If you give it, what will happen is it can go to any other cell and destroy any good cell, isn't it? And therefore there is a lot of contraindication, isn't it? But in this kind of targeted therapy where toxin and engineered antibody can be given, it becomes very, very specific, and therefore it will help you a lot. On the other hand, there are several other examples as well. For example, if you see interleukin 2, then EPO, erythropoietin, again a very important example in what we call as engineered protein. Erythropoietin is very, very essential for maturation of RBCs. Okay, now those patients who have not got rid of their kidneys due to renal failure and all such things, what will happen is they will not able to produce EPO or erythropoietin all by themselves, and therefore an engineered EPO should be delivered. Okay, now this engineered antibody, this protein is nothing but a recombinant DNA generated protein, and therefore it comes in a very cheap price, which is will be not possible if you really isolate EPO from human beings. Similar is the example of a very good and very common hormone, that is insulin. Now today, in order to tackle the problem of hypoglycemia in patients, that is diabetes mellitus, so what you do is the ultimate therapy is insulin. Now insulin is a protein and it is made by human beings only. So if you take insulin from one human being and then purify it, it becomes hugely costly and it will not be this thing by anybody. So in that case also, we can what we do is we can use recombinant DNA technology eventually to make this insulin. And this is also true. Once upon a time, all in the beginning, it was not possible because at that point of time, genetic engineering was not available. At that point of time, what the scientists used to do is they used to procure in from pig pancreas. But there was a lot of problem to use the pig pancreas because it will cause a long-term what we call is antigenic response in the animal, and eventually the patient will get rid of hyperglycemia, but the patient eventually may die due to long-term what we call is hyperimmunity. Okay? So in order to reduce this kind of conditions, recombinant DNA technology has been sufficiently used. One more very important example that I must discuss is interferon gamma, IFNG. Interferon gamma is being used for antiviral therapy. In India, PGI Chandigarh has been in the forefront to use interferon therapy for hepatitis, all kinds of DNA viruses. That is hepatitis B, C, and the possible all possible varieties of hepatitis viruses. So there has been excellent result, and it has been standard use interferon gamma for hepatitis. So this is also then there are some other uncommon examples which only a person with specific interest will able to understand, but several other examples are also there. Okay, I'm not exactly discussing this. So and one more group of peptide based this thing drugs, enzymes and catalytic proteins. Okay, this is very common in pharmaceutical industry as well. So in Indian context, if we say that because the Indian food habit is not very good, I say, so what happens, they suffer a lot of dyspepsia and lack of absorption and malabsorption syndrome and things like that. So in order to control this, what you need to do is you need to have your enzymes and proteins in a proper diet. Okay, so because your pancreas is not very strong to produce all these enzymes, you take these supplements. So if you go to any shop, you ask for some enzyme, they'll give you with some pineapple flavored this thing enzyme, and several other things are also there. These are also peptides and these are also drugs, and therefore it's a very big this thing what we call industry where enzymes and catalytic proteins are being used for therapeutic purpose. Now the production of this enzyme and catalytic proteins are also very easy because most of the time you use only solid state fermentation to produce this by using a particular fungi cell. Understood. In this way, what will happen is you can bring about very good opportunity in pharmaceutical industry as well by means of recombinant DNA technology and all such protein engineering that we are interested in. Now, this is again one more important aspect of production of vaccines. So, I'm giving you an example where in 1998 you started about thinking about the enzyme vaccine, and eventually at 2008 and 2010 you end up with the vaccine after the preclinical trials 1, 2, and 3. Understood. So what I'm trying to say is here it needed approximately 10 years to make a vaccine one day, but today you can generate a vaccine within 10 years, within 1 year only. Just hold on. হ্যাঁ এখনো হয়নি, আমি পরে কথা বলছি। So, am I audible, Professor?
Yes, you are.
And the screen is visible also?
Yes, sir.
Okay.
You are audible.
Yes, sir. Yes, sir.
Fine, absolutely fine. So what has happened is this is a timeline which is usually needed to develop a vaccine, which usually takes 10 years, but due to rapid development in various aspects that I will discuss today, and we have had an opportunity to develop vaccine within a year. And this is true. All you have taken 2 jabs of vaccines, and within 1.5 years only it has been successful. So we shall see what exactly has happened. So these are some other considerations that you should take when you are using a peptide-based therapeutics. Some examples I will discuss. For example, biocompatibility. There should be sufficient biocompatibility, and then antigenicity. Now, antigenicity should be reduced if you are taking it for some other purpose. But if it is a vaccine, then antigenicity should be increased. So there are different aspects to it. I'm not going to discuss it in all detail, which needs a lot of discussion. Then there is one more important aspect, that is cross signaling. Now, cross signaling is something like when you are talking about growth factors, as I have given you an example, a patient who is suffering from bed ulcers, okay, bed sores. So you are giving him a fibroblast growth factor or epidermal growth factor. Why? In order to repair his muscles and skin, okay. Now these epidermal growth factors and fibroblast growth factors are also needed by cancer cells. Okay, now in cancer, what will happen? A lot of cells will develop rapidly. Okay, so in that case, also they will use this kind of growth factors. So you should be aware of this kind of crosstalk. Understood? Molecular level crosstalk when you are applying something and you are getting result for something else. So these kind of things also should be needed to taken care of when you are using peptides and proteins as a drug. Okay, I do not know how you have understood, but it's a long process and very much intricate mechanisms are there which the author or the scientist should be aware of. ऐसा नहीं होना चाहिए आपने एक ड्रग दे दिया उसके बाद इसका क्या हुआ उसका पता नहीं और ये क्यों हो गया इसका भी पता नहीं ऐसा नहीं होना चाहिए. Therefore, a lot of study should be done and accordingly you should go ahead. So these are some very good examples where protein engineering has been drastically used and has been done excellent work. Now in Eastern European countries, mostly Western European countries, mostly Scandinavian countries, this protein engineering is very rapid and they are doing excellent work there. Examples like companies like Eli Lilly and then Novo Nordics. These are all Western European these companies which use a lot of protein engineering for development of therapeutic peptides. Okay, so I could have given you some more examples, but it would be out of scope. So it is the number of patents that they have filed, and as you can see, Scandinavian countries, and then USA, then Japan. These are the countries who are forerunner in this. So there is a lot of scope in India as well. So this is how an enzyme works. So most of us are teachers. In order to teach, it's a very good slide which directly shows that how catalytic proteins will work by reduction of the G for a particular reaction pathway. So I'm not discussing it either. So the one more problem here is how you will stabilize the peptides. Okay, the stability of the peptide is also very important. Let me give you one example. For example, you have made a drug with using a particular peptide. Okay, now the shelf life of the peptide is so small. तो क्या होगा आपका उसको बोलते हैं एक्सपायरी डेट. एक्सपायरी डेट बहुत करीब होगा. मैन्युफैक्चर किया और जल्दी जल्दी एक्सपायर हो गया तो कंपनी का तो फायदा नहीं होगा. सो कंपनी क्या चाहेगा? कंपनी चाहेगा कि जितना मतलब ज्यादा हो सके उसका एक्सपायरी डेट बढ़ाया जाए. सो हाउ एग्जैक्टली यू विल डू इट? So you have to do some special mechanism so that the stability of the peptide is enhanced. So in order to stabilize the enhance the stability of the peptide, what are the things you can do? You can change the delivery mechanism. You can even immobilize the protein so that it gets entrapped in something and then remains stable because the proteins are very fragile, isn't it? It is a biomolecule and you need to preserve it. आप भी देखे होंगे कि बहुत सारा मेडिसिन है जिसको फ्रिज में रखना पड़ता है. Any medicine you are keeping in fridge, you would be sure that there are some peptides. Okay, peptides is nothing but a protein. Okay, your body, your blood, everything is made up of protein. I hope most of are from biotechnology and this kind of background, they know these things. But those who are from a different background, because ATAL has gives you an opportunity where interdisciplinary talk is possible, and ATAL promotes this kind of interdisciplinary discussions. Okay, so what will happen is need is also like this. So if you are very much classical, that you are doing only computer science, so your horizon becomes limited. So there is no this thing in basic research and basic science. There is absolutely not. But at the same time, interdisciplinary aspects of doing work together is also very important. And therefore, ATAL has given an this thing opportunity where computer scientists, mechanical engineers, and biomedical scientists all can discuss. Anyway, one more thing is peptide assembly. By peptide assembly, so you are increasing the overall molecular weight of a small peptide. I'll give you a specific example of our lab also, how exactly we have achieved it. Anyway, these are some delivery mechanisms by which you can reduce the degradation of the peptide. Understood? For example, the COVID vaccine. In COVID vaccine, you need to make it in a particular preserve. This thing you cannot use it like simple like oral polio vaccine. The polio vaccine is also vaccine, and COVID vaccine is also vaccine. COVID vaccine needs to be preserved in a particular way because it is very fragile and you cannot play with it. On the contrary, the polio vaccine is very highly stable, and therefore, if something goes wrong, also the activity remains, and it is also very excellent. Until now, there has been very good results of polio vaccine, but in case of COVID vaccine, this and the results are not very good because, as you know, after taking 2 jabs, also you are not completely what you call is immune from this particular disease. Anyway, in the meantime, since these are post-lunch sessions. And most of the people will feel dizzy and not able to concentrate. So let us watch some videos. So I'll request this thing, what you call is our coordinator sir to play the first video in mute condition and I shall speak.
Okay, yes.
In mute condition, in full screen, yes, absolutely fine. Now this is the whole of production of insulin. Okay. Now insulin is getting produced inside an organ called pancreas. Now inside the pancreas, the gene for the production of the insulin is there. Okay. Now gene is nothing but the hard copy, where from where you will make multiple copies and then finally you will get in. Okay, by some several process. Okay. The blue color cells are those produce insulin. Okay. So inside the blue color cell you will DNA. Okay. Now inside the DNA, the lot many more information is there. Okay. It is not that this inside this blue color cell only gene for insulin is there. Lot many other genes are also there. But you need to get out what exactly is it and have to isolate that particular gene. Okay. So they'll show you how. Now this is the whole gene. Okay. This is the whole gene. From the whole genome, it's like a book. From the book, only 2 pages are needed. So you will take a xerox of 2 pages only, isn't it? So you have to find out what exactly the 2 pages are. What the codes for insulin is then. Just hold on. In order to do so, by a specific mechanism, by using genetic engineering, we will isolate the particular vertical DNA. Okay, the genome, the whole DNA, and from that a small part of it will. Okay, see, these are some molecular scissors which will cut the DNA into small pieces, and this particular piece is production of insulin. Okay, okay, it is getting it. One more cartridge. Okay. Now this is the small part of it. Only this much is there that for insulin production. Okay. Now we are putting it into a expression vector. An expression vector is also only in a different form. It is a circular form. Okay. Now what is we have not taken the genome but small part of it and putting it in host cell. Now this host cell can be an E. coli cell also, a common E. coli cell. Now the E. coli cell will not understand what is going on. Producing the protein. Okay. Instead of the fact that this protein is not needed by E. coli but by human. E. coli will not know, and eventually insulin will be produced in the medium where E. coli is growing. Okay. Then eventually what you will do is you will take the whole DNA and put it into the bad cell, and what will happen is previously the cell which was not able to produce insulin able to produce insulin. Okay. Now this is how a recombinant protein is being made. Okay. Fine. So it's a very simple representation. Sir, would you please share the second video?
Yeah, yes.
So what all the objective is to show this thing that drug engineering is have plenty of other multiple this thing where you should engineer drugs, but there is a whole lot of drugs which are made peptide, and peptide engineering is also an important aspect to. You can have excellent results and very good. Okay. No, right now I don't want it. I thought that some other video is there. Okay, sir. Sir, I'll go to my presentation and later on I'll come back. Not needed, sir, now. Okay. Okay. So what has happened is, as you have seen, so the problem with peptide is what? It is very unstable. So in order to increase the stability, what you will do? So you put it in a liposome. Okay. Liposome is nothing but a membrane which is made up of lipid. Okay. Now inside the lipid, the protein will be preserved. Therefore, it will have much more what they call action. Understood? So in this one, what will happen is, if you see it properly, that the protein is embedded inside. So I'll give you a specific example also. So all of you are aware of vitamin D, isn't it? Now vitamin D is very unstable. So when you go to a shop and ask for vitamin D and calcium capsules, they'll give you. In that, approximately 60,000 international units of vitamin D will be present. Now, there is a problem with the vitamin D. What happens is it's very unstable, and therefore it may be a possibility that you end up with only 1,000 international unit of vitamin D instead of 60,000. That is the dose that is needed. Why is this? Because it undergoes rapid degradation. So the company has made this thing production of vitamin D, where they are isolating vitamin D from salmon. Now, salmon is a fish which is present in European countries. So what they do is the salmon vitamin D is also very unstable. So what they do is they put it in a liposome and make into a nasal spray. I don't know how many of you, many persons who is having older father and mother, usually doctor is giving this particular medicine. So it's a nasal spray of vitamin D. Understood. Now this nasal spray, in that nasal spray, vitamin D is embedded in a what do you call as a liposome, and then the liposome is getting sprayed like an aerosol, okay, and you will inhale it and your vitamin D will be delivered. So it is a different from the route of delivery of usual vitamin D. So this is the usual process by which we will prepare this what it is liposome. Understood. So this is one more strategy where we can enhance the stability of the peptide. As you can see, embedding in a 5 hydrogel and then cross linking, then copolymerization. These are nothing but the methods by which you can increase the stability of a peptide. Okay, and the ultimate goal is what? Ultimate goal is to give addition compliance. Okay, for drug engineering of all sorts of drug engineering. Ultimate goal is like this only. So this is one more where you are preserving catalytic pockets. If you see nicely, you can see the catalytic pockets are green colors. Can you see? These are the enzymes, and in between it is like half moon, isn't it? The open mouth you should preserve because this is your active site. So how you will preserve? You will add something which will stabilize the active site, and what you will have is you will have much more stable enzymes. Understood? So overall objective is what? Overall objective is to give a high stable proteins. Now this is one of our reports. I think we have published it in 2017. So we worked here for 2-3 years and we isolated some peptides. Now these are the bioactive peptides, okay, which was been isolated in our lab. Now the molecular weight of these peptides are very, very small. You understand one thing? We isolated the peptide in pure form and we saw that it's highly anti-pathogenic, okay. Very important pathogens are getting rid of this. But the problem was this, that the stability of the peptide is very low. Once the stability of the peptide is very low, you have to devise a strategy of stability of peptide. Now, how you can do it? Among several methods that we already discussed, one more method is there, which is artificially increasing the molecular weight. So, what exactly you will do here? What usually happens is in enzymes and proteins, the active site is very, very important, as you already know. Now, in order to preserve the active site, there is a mechanism which is called this thing. What we call is now steric hindrance is a mechanism where some other molecules will make a barrier to the internal part of the enzyme and the outside environment. Now, outside environment is very, very dynamic, isn't it? So much of ionic changes, then interactions, hydrophilicity, hydrophic interactions. So many interactions are going on everywhere in this thing. But you need to preserve your active site from these interactions. So what you do, you artificially make so many molecules outside it, and therefore you make a barrier between your internal active site and the external thing. So in order to protect your internal units, you can use some exterior molecules. Now once you use exterior molecules, protein will become more antigenic because you are using more than one peptide. So what is our strategy is we will use the same molecule only as a scaffold, as a peptide, as an assembly. So what will happen? Many of the protein molecules site interior to the core will remain active. Okay. So in order to do so, we need to do a lot of hard work. For example, we have had a collaboration that is still in talk with this Cavendish lab of Cambridge University, who are the fathers of peptide assembly and all the possible usage. Now this lab, also the lab which have already told about COVID vaccine using a nanopeptide during 2012. So you can think about this thing. They at a point of time only that one day it will come that the coronavirus will be a very bad pandemic through the world. At that point of time, it was COVID-19 because it was not discovered at that point of time. It was the same coronavirus only in a different form, and they used to predict that it will be a huge menace in the long term to humankind. They proposed about a peptide-based vaccine at that point of time only, 2012. You can even search it in the internet, and you can read the full report. Anyway, now what is this thing of thermodynamic stability of a peptide? Please understand that all kinds of reactions governed by the thermodynamic equilibrium, which says that the delta G is inversely proportional to the what we call as degree of disorder, or we shall say entropy, or delta S. Okay. Now what happens is, if you want to make some unstable peptide into a stable peptide, what you need to do is you need to reduce the degree of disorder. So eventually you need to reduce the magnitude of delta G, isn't it? So it is also a very good representation here, where you can see that if you reduce delta G, what will happen? The molecule will become more and more stable, and it will become ice. Understood. So what we are doing is we are trying to reduce the delta G into negative magnitude, and therefore we are trying to make an artificial adjustment so that the stability of the peptide is enhanced. Okay. So when you are putting something in fridge, you are also doing the same thing only, but we are not putting it into a fridge because the fridge is costly. We want it to make it room temperature only, but it will be engineered in such a way that its stability will be increased. I hope I have made you understand what exactly is going in the whole discussion process. Now, in order to do so, what we can do now, this is the small peptide that we isolated, okay, in our lab, that we have reported in 2015, 16 something. So what has happened is, this is a small lab with only, small peptide with only 9 amino acids. Understood. So it is very, very highly unstable because the molecular weight is very low. So what we did is we assembled into, okay, we assembled into a this thing. This is the dipeptide, and later on the tetrapeptide. Yeah, this is the duplex peptide, and later on this is the tripeptide. Now, when we achieved the tripeptide assembly, we found out that the stability has also been increased. How we calculated? Now, you see here. This is the initial peptide stability, where the last column, if you see, it is minus 150 approximately kilojoules per mole. Okay. Now, when we did the assembly to the tripeptide, the overall this thing delta G has reduced to a significant extent, approximately minus 300 kilojoules per mole, if you can see. So what happens is, and we simulated also the whole process, isn't it? And so by specific mechanism that I will discuss later on. So what we understood is, by enhancing this thing, what we call artificial molecular weight, we can do is we can stabilize the peptide. Okay. So as you can see here, the topmost one is the tripeptide, where the stability has been significantly reduced. Okay. And the bottom one is the single peptide, where the stability is very, very high, as you can see here, from something called what we call as R square, or root mean square of displacement. Okay, this is a concept which is a thermodynamic concept only, where we are calculating the mobility of each and every atom that is present in a protein. Okay, by a concept called Brownian molecular dynamics simulation. So by this we have adjusted, and we have seen that the overall stability can be increased by putting it into a matrix. Okay. So there are some other this thing also, some representation also. These are not made by our lab, but somebody else. So here what is happening is, you anticipate to make one kind of product, okay, but you are not getting it. Why you are not getting it? Because you do not have a specific kind of enzyme. The enzyme you are having. So I give you an example. So see here, the substrate here is a candy in which blue and red color. And my anticipated goal is a candy which is completely blue in color and completely red in color. Okay. So in order to do so, we will add some enzyme. Now the enzyme is such that it is breaking down the whole candy. Can you see? Small red and blue boxes. I don't want that. So why it is happening? Because I do not have the specific kind of enzyme that is needed for to do this particular action. So in order to do so, what we will readjust the structure of the enzyme and change the what they call function of the enzyme completely, and therefore ultimately we can have our anticipated goal. Okay. Now this is the concept of whole this thing, protein engineering. Okay. Now how exactly we will do that? So there are a couple of examples where protein engineering has given excellent result. For example, in the first column, if you see, there has been a 52-fold increase in the output of a particular enzyme by a single genetic modification. So we shall see some more example. Here you see, here we have changed the particular lab who have proposed it, and they have completely changed the substrate specificity of an enzyme. So initially in the wild type, the enzyme was only accommodating a proton. Later on, when you made into a mutated protein, Understood. Mutated enzyme. Now it can accommodate a whole lot of big molecule, that is benzaldehyde, and therefore it can produce a different kind of this thing also, molecule also, isn't it? So this is how what is happening is it is completely changing the this thing ability of the enzyme to do some work, isn't it? So eventually there are several other this thing also. This thing what you call is this thing what you call activities also where you are not you are changing the complete structure of the protein, and therefore you are like ঠিক আছে চল. So what they'll do is they'll completely change the output of the enzyme as well. Understood. So this is one more example where previously the substrate that was given was 3 malonyl CoA, and rapidly when we have changed the structure of the enzyme, the ability to accommodate 2 malonyl CoA has been accommodated. Now why is this? Because initially the active site of the protein was large. Okay, so after some protein engineering, we have changed the structure of the active site, and eventually 2 malonyl CoA can be accommodated. Okay. And how exactly we will do this? We'll put it into a process called error-prone PCR. Okay. In the error-prone PCR, what happens is we change the structure of the gene in a complete different way, and eventually we end up with so many mutants. Out of all the mutants, you have to screen which exact mutant do you want, and eventually we can find out the specific mutant that is there that can be used. So error-prone PCR is very much in use, and eventually you have to identify the whole the mutants which will help you to or do a complete shuffling of the kind of enzyme that is anticipated by you for your work. So this is again a PCR based method through which we can eventually generate mutants, and eventually a specific screening strategy should be there. I'm not going to discuss it in a complete elaborated way because it would be only interest for some specific labs. So one more important aspect is which kind of host cells you are using. So this is also very important. In the video they have shown E. coli, you can use something else, but it depends exactly which kind of protein you are preparing. Okay, this is again a PCR based method where you can able to introduce mutations in a DNA. These are some examples where in starch processing industry, because starch processing industry is fundamental to all many kinds of industries, including textile, drugs, pharmaceuticals, and so on. Therefore, some specific examples here where protein engineering has given excellent results. Okay, so and this ability to simulate is also there because now we can handle a lot more data by using machine learning and tools like this. So we'll discuss a specific kind of machine learning tool through which we can like exploit protein engineering, not in the this thing what we call wet lab, but in the dry lab as well by means of simulation. I'll discuss it, but before that, may I request our coordinator to please share the links that you provided. Please tell me what are the links we have given, sir. Other than the video links, is there any more links we have provided to you?
You have provided, sir.
What are those?
One is about
simulation link as we have given.
Yes, simulation software link is there, and two models are there. L1 model. Shall I share this?
After the video links we read like youtube.com like this. Okay. The simulation links, can you read it out so that I can understand which exact simulation we have provided?
Yes. Yes.
एक बार सर क्या लिखा? हाँ.
Yes. It's a L1 model ASP74PH.
हाँ?
L1 model domain.
और एक बार पढ़िए सर.
and one model.
नहीं ये नहीं, लिंक 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 HTML लिंक नहीं,
Yes, yes, yes.
ये तो डेटा फाइल हो गया,
Okay.
ये लिंक नहीं है, मैं लिंक के बारे में बोल रहा हूँ.
Yes, yes.
Sir, shall I share it? Shall I share that screenshot?
No, okay, no problem, please share.
Yeah,
That will be better.
I will be sharing. Yeah. Wait.
स्क्रीनशॉट एक बार शेयर कर दीजिए सर.
Yes, yes. My screen is visible.
No, I'm not asking this. Just hold on, sir. You close it. Can you please, if you don't mind, open the email so that I can read which are the links I have shared with you,
Okay, sir. Just
or only the video. Please, just you open the email. Just one
that
second.
email. Yes, I opened that.
There are two simulations, sir. Simulation 1 and simulation 2. Have you seen it?
Yes,
NC
sir.
बोल के है, देखिए.
Yes, sir. Yes,
और
sir.
इसके बारे में बोल रहे हैं सर, इसके बारे में बोल रहे हैं. अच्छा रुक जाइए, क्लिक मत कीजिएगा.
Yes, sir.
रुक जाइए.
Tools open करना है.
मैं बोल रहा हूँ. नहीं, सुन लीजिए. इसको सिलेक्ट कीजिए, सिलेक्ट कीजिए, कॉपी करके पहले कॉपी कीजिए सिर्फ लिंक को, सिर्फ एक नंबर का लिंक को. नहीं, क्लिक तो बाद में करेंगे. कॉपी कर लीजिए सर एक बार, और पार्टिसिपेंट्स को भी शेयर कर दीजिए उनके चैट बॉक्स में. हाँ, अभी ठीक है, अभी कॉपी कीजिए. हाँ, ये चैट बॉक्स में ताकि वो भी एंटर कर सके, वरना मैं ही मतलब करूंगा, ये लोग तो नहीं कर पाएंगे. पार्टिसिपेंट्स को शेयर कर दीजिए उनका चैट बॉक्स में. ये अगर चाहते हैं तो वो भी कर सकते हैं, हम जो अभी सिमुलेशन करने वाले हैं उसको.
शेयर कर दिया. Yes.
हाँ ठीक है. ओके. हाँ, अभी आप आपका स्क्रीन शेयर करके रखिए. हाँ, बहुत बढ़िया. अभी आप आपका स्क्रीन को शेयर करके रखिए सर. हाँ. सो हाँ, नॉट दिस द प्रीवियस. हाँ, ये वाला लिंक को शेयर कीजिए सर. क्लिक कीजिए सर इसको अभी. हाँ, इस पर क्लिक कीजिए. हाँ, ये वाला ऊपर वाला को इसको बात दीजिएगा. इसको एक बार खत्म कर लेते हैं सर. हाँ, एक बार इसको क्लिक कर लीजिए.
Yes, sir.
हाँ सर, बहुत बढ़िया. Okay, so dear participants, so what happened is, as I have said, that we can do the dry run also. That kind of question we can do. Understood? Consider that you are having an enzyme and its stability is very low, or one of its properties is not very good. You want to change it. So how you can do it? You can do the mutagenesis as we have shown you, which requires a lot of reagents, equipments, and skill, isn't it? So अगर ऐसा हुआ कि आपने एक एक्सपेरिमेंट कर लिया, तो उससे बहुत सारा इन्वेस्टमेंट भी कर, रिजल्ट आपका ठीक नहीं आया, तो इट विल बी वेरी डिमोटिवेटिंग. So beforehand you need to do some simulation so that it becomes this thing, and you predict that which will be good or. So in order to do so, what we'll do is we'll use some data files. Okay, so this is the first data which will give you data of genomes, animal and plant genomes. So I'm not going to do any simulation right away, but it is a database which is there to find out any. For example, हमने एक वीडियो देखा था, उसमें from the whole genome a small part of the genome has been isolated. Can you remember? इंसुलिन के जीन के लिए. ठीक है, तो इसका भी हम ऐसे ही यूज कर सकते हैं. We will not do in this thing what you call wet lab, but in dry lab we can eventually do it. Okay. Now this was the first thing that I wanted to share with you. Now it's a genome database. Now from this genome you can find out all genome database. You can find out genomes of approximately many animals, plants, as well as bacteria and all such things. ठीक है. Later on, the second link is also there. Sir, go to the second link from the email, and you can also share with your participants if they want.
First I will share it.
इसको भी एक बार थोड़ा शेयर कर दीजिए. हाँ, और उसके बाद क्लिक भी कर दीजिएगा सर. एक बार शेयर कर दीजिए लिंक. हाँ, if some persons are interested to see how exactly these things work, they can definitely do it. अच्छा, now we can fine. Thank you, sir. Okay.
Yes.
So please see, as I have said, मैंने एक बार आपको बोला था कि in electrodes, that means in biosensors, for example for blood glucose का सेंसर है, देखिएगा बहुत सारा कंपनी का मिलता है. You take a small drop of blood and put into the electrode, and eventually you'll find out how much glucose is there, isn't it? All these electrode use this kind of enzyme, which is cytochrome oxidase. Now for cytochrome oxidase also, from the whole genome, we can find out a small part. Now this thing we done. Understood, right? Now we did a simulation where from whole genome, we have taken a small part of it, okay? Now this is a small part of it. We usually have the Pseudomonas. Pseudomonas is a bacteria from where cytochrome oxidase is usually isolated. Now Pseudomonas का whole genome का sequence भी है पहला वाला database में और उसका एक part भी है, like this is a small part of the whole genome, okay? Now this particular database in which the whole genome is there, as well as the bulk of the cytochrome oxidase protein is also there. This particular format is called GenPept format, okay? Now this GenPept format is such that so much of information is there, बहुत सारा information है, ठीक है? इतना सारा information का overload है कि कभी-कभी ये दिक्कत हो जाता है. So in order to make it more useful, we can convert the format also into a very simple format, okay? So in order to do so, sir, may I request you to please click on FASTA. FASTA, if you can see. FASTA blue color, if you can see, please click on it,
Yes,
sir.
sir.
हाँ, बहुत बढ़िया. इसको click कीजिए, sir. हाँ. Once you click on it, what will happen? All the unwanted information will go, and only the protein sequence will come. Can you see? It starts with a greater than sign, and then there is a unique ID, then the name of the enzyme, then the name of the bacteria, and then we are having the sequence. Can you see? M, R, I, G. Can you see? Each and every letter is nothing but a amino acid code. Okay. So M stands for methionine, I stands for isoleucine, R for aspargine, and so on. So these are the different amino acids that is present in the protein cytochrome oxidase. Now we'll do how you'll see that how cytochrome oxidase will be changing its structure and stability. Okay. For that we will do the second simulation. Okay. So may I now request, sir, please to click on your desktop and the icon that you have downloaded, that is Rasmol.
Yes, sir.
So minimize it, sir. We will see the simulation how it looks like. So please click on the Rasmol. This thing. Now you share your desktop and please click on the Rasmol icon. Yes, and rest of the participants can also do. So what we are doing is we are taking from a big database of cytochrome oxidase, we have isolated. Now we want to make it into a protein, exactly how it looks, and how will you determine that which one is good protein and bad protein. That also we'll see. So, sir, first of all, please, no, don't open, sir. Please, first of all, please click the icon. इसको भी आप क्लोज कर दीजिए.
Yes, sir.
सिर्फ डेस्कटॉप दिखे ताकि एग्जैक्टली हमारे पार्टिसिपेंट जो हैं वो कर सके.
Yes, sir.
Many are already aware of it. हाँ, प्लीज क्लिक ऑन इट. डबल क्लिक कर लीजिए सर.
Yes.
तो विंडो ओपन होगा.
Yes, window opened. Two windows are there. One is command window.
But it is not visible. I don't. No, command window को काट दीजिए. हाँ. What happens is in the software there are two parts. One is the GUI, graphical user interface, and then the command line. So
Yes.
it will take much time to learn, but this is very easy to understand. Okay. Now what I will request you is
Yes.
you minimize this window. ये छोड़ कर लीजिए window को थोड़ा छोटा कीजिए. Close मत कीजिए, छोटा कीजिए.
Okay. Just
इसको क्लोज कर दीजिए. इसको क्लोज कर दीजिए. मेरे को लगता है आप जो शेयर कर रहे हैं वो स्क्रीन अगर हाँ ये ठीक है. और दूसरा टाइम और एक बार आप हाँ इसको भी क्लोज कर दीजिए.
वो क्लोज हो जाता है.
दूसरा टाइम और एक बार आप आइकन को स्विच कीजिए. नहीं हाँ. व्हाट आई वांट इज दिस काइंड. हाँ ये वाला जो नहीं बड़ा मत कीजिए, रखिए. ऐसा दो विंडो ओपन कीजिए सर.
दो विंडो ओपन करना.
मैं शायद समझा नहीं पा. हाँ
Yes.
टू विंडो के साथ ओपन कीजिए. हाँ
I opened that two windows.
टू विंडो ओपन हो गया. ठीक है. नाउ दिस प्रोग्राम Rasmol is written in C++ and Python. Okay.
Yes.
It's a very small package. So two windows are opened of the same software. Okay. So participants can also do the same. हमने क्या किया है, हमने पूरा डेटा देखा है जीनोम का, उससे एक प्रोटीन का पार्ट लिया है साइटोक्रोम ऑक्सीडेज. ना उसको भी स्ट्रक्चर में डाला है. ठीक. अभी स्ट्रक्चर में कौन सा वाला प्रोटीन अच्छा है वो वाला भी देखेंगे. एक करेंगे म्यूटेंट वाला प्रोटीन, एक देखेंगे वाइल्ड टाइप प्रोटीन. वाइल्ड टाइप मतलब क्या, नेचर में जो है ना. नेचर में उसका एक प्रॉब्लम, क्या प्रॉब्लम है इससे अगर हम इलेक्ट्रोड बनाएंगे, इलेक्ट्रोड का एक्सपायरी डेट 1 मंथ होगा. कंपनी को नहीं, तो कंपनी क्या करेगा, ऐसा इलेक्ट्रोड बनाएगा जो 1 साल तक चले. इन ऑर्डर टू डू सो यू नीड टू स्टेबलाइज द प्रोटीन. ओके. नाउ व्हिच प्रोटीन, cytochrome oxidase. So we'll see after stabilization how it looks and before stabilization how it looks. And we also would like to understand that why the stability has been changed. Okay, so in order to do so, we have downloaded a software that is called Rasmol, and the two windows subsequently we have opened, and we have also shared with you two data files. Now in the first window, go to the file option, sir. In the first window, go to file, open, open, uh, and click on the first data file. Open it. ओपन इट हाँ हाँ अब इसको छोड़ दीजिए, कुछ मत कीजिए, इसको हटाइएगा नहीं, अब दूसरा वाला विंडो पे चला जाइए।
ओके ओके,
दूसरा वाला विंडो पे चला जाइए, फाइल में जाइए, फिर से दूसरा वाला,
यस,
यू प्लीज गो टू द सेकंड विंडो, गो टू अगेन फाइल, ओपन एंड पुट एंड आर यू श्योर इट इज द सेकंड दिस थिंग?
यस सर, इट इज सेकंड वन, यस
सेकंड फाइल ना, ओके फाइन फाइन सर, इसको मूव मत कीजिए, प्लीज डू नॉट मूव इट।
हाँ यस,
हाँ और एक बार कर लीजिए, हाँ हाँ अभी ठीक है। Now here you see participants are exactly. So what has happened, participants, as you can see, both the proteins now in its this thing they call skeleton mode. Okay. Now in skeleton mode, what is exactly happened? Only the C alpha bonds are being shown, peptide bonds. Okay. But now we'll put the atoms on it. Sir, please go to the display window and click on it. अभी molecular surface एकदम bottom में, लास्ट में देखिए मॉलिक्यूलर सरफेस होगा, हाँ इसको क्लिक कीजिए। अभी इसमें चले आइए, इसको होने दीजिए, हाँ इसमें चले आइए, दूसरा वाला पेन में चले आइए, यहाँ पे भी सेम ही कीजिए, डिस्प्ले मॉलिक्यूलर सरफेस।
यस यस,
great. So these are the two cytochrome oxidase which are equal in all respect, not exactly all but most respect. Okay, as you can also see, the pattern is also similar, isn't it? Now out of these two, one is a white protein and another is a mutated protein. ठीक है, so mutated protein ताकि उसका स्टेबिलिटी बढ़ सके, ताकि हम जो इलेक्ट्रोड बनाए वो 1 साल तक अच्छा रहे। ठीक है, so how we will understand which one is mutant and which one is exterior structure is similar, not exactly same but mostly similar. So we'll do is we'll go to color option. Please go to color option, sir. Please go color option. Color. इसमें देखिए टेम्परेचर एक ऑप्शन है, सब मेनू टेम्परेचर, यू गो यू क्लिक ऑन इट। ओके, यू गो टू द सेकंड प्रोटीन टेम्परेचर। ओके ओके, नाउ एग्जैक्टली व्हाट इज इन आवर फ्रंट having a plane both where each and every delta G of each and every atom is been represented. Okay, now according to Rasmussen, if the delta G is more than less than minus 100 kilojoules per mole, the color of the atom will become blue. Please understand, the software that is Rasmol is been instructed in such a way that if the delta G of any molecule becomes less than 100 kilodalton, the color will by default become blue. Okay, now this is true for all any Rasmol package. Okay, now we did not do any change in the software but only the structure of the data file. Okay, now right side data file which is completely blue with a small red nose, it is the mutant. This mutant is a single mutant, that means a point mutation has been done to it. Okay, but the previous one is a wild type. In wild type you see different kinds of colors are being shown. Red is highly stable and blue is most stable, and the whole protein is such that different areas have been different stability. Okay, but in the right side it is completely blue and a small part is red. So what does it mean? That the mutant is highly stable, and if we get rid of the red nose, it will become very stable. It will do rest of the other things same but with high stability. Are you getting? So this is how we putting protein engineering for therapeutic purpose, and just giving you this thing how to enhance stability. So by protein engineering and by mutagenesis, what we can do is we can completely convert the structure of the protein, and eventually we can generate very good mutants, isn't it? We can generate a mutant library, and eventually we can finalize which mutation is good, and then you can go for the wet lab experiments. Okay. Okay. So thank you, coordinator sir, for your excellent coordination, and I will now go to my this thing presentation. Okay. So what has happened is just no. Okay. So in to think that there are how the software. Okay, now this is the protocol how the software will find out the pattern, about the tools for use. Now this is also useful to understand the vaccine design and why the mutants are not being read of by after taking two vaccines. So there are several aspects to it, and we reported this in recent and this particular also in which we discussed about the mutations in COVID. What the mutation in 2021 the mutants. Also in the action tool of this whole of genomics to do the induction. So this is the whole genome. The Pseudomonas whole genome small part of cytochrome oxidase, isn't it? So we used for as well. In 2015, we they started this product. So by the finance or finance so it will hold with integrated parent and we are with the to smooth complete
Can you please check? Dr. Sujit, your voice is not clear. Can you please check?
Went to motivated by small scale industrial. So being said so, to conclude my session, if answer some of your questions, I'll be very happy. So, coordinator sir, my voice is
Sir, your voice is not clear. There is too much noise.
Dr. Sujit, your voice is not clear. Hello.
Am I audible now?
Yes, yes.
Hello.
Clear. Yes, 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 yes.
Okay. So this was the idea that how protein engineering can also help for drug design and drug engineering. That was the theme of your FDP, and I'm very much thankful to the coordinators that they have proposed this theme and given an opportunity to me to interact with the participants. So if there are some specific queries, I would like to answer some of them. If I can able to answer, I'll be very happy.
That is done.
Am I audible, sir?
Yes, you are clearly audible.
Okay. Because drugs are such that it is not only small molecules and salts. Usually, when we talk about drugs, we are essentially thinking about some small molecules like paracetamol or salts like amoxicillin and things like that. Other than that, also the peptide also essentially can be a very good drug, isn't it? If it can alleviate some symptoms in a patient. So in that case, when we talk about drug engineering, we essentially should put the therapeutic perspective in peptides also, proteins also. So that was the objective of my session, to discuss and to also talk about some specific things that are going on recently. So if there are some specific queries from our participants.
You can type it also. Participant can type the query.
They can also write it down in the chat box if they want. Sir, can you please describe biomarkers? Oh, excellent. Yes, sure. Why not? So biomarkers are those which can identify some specific kind of ailment that you are going through. Okay, let me give you some very normal examples. For example, there is a thing called ASO, anti-streptolysin O. So in blood, if it is more than the normal range, it indicates that you are suffering from an infection caused by Streptococcus bacteria. Okay, so there are plenty like this. Biomarkers are something which will give you an indication that what is what is the normal condition of the body, whether it is normal or not. If it is going on wrong, what exactly is going on wrong? Okay, some more. If we say, for example, like anti-insulin antibody. So if you are having more than a normal anti-insulin antibody, it means that your insulin molecules are not working properly because there is an antigen-antibody interaction. Okay, so in this way, a biomarker can be something which will give you an idea that what exactly is going on. Now you said one more thing, I think, biosensors. Okay. Now biosensors are something which will give you the quantitative thing of the exact the quantity of the particular metabolite you are looking into. For example, glucose. So you must have been aware of this glucose electrode that is there in the market. So what exactly are these? These are nothing but biosensors only. What they do, they identify something which is biological in nature. So there are some more examples as well. For example, an ECG electrode is also a biosensor only. Why it is a biosensor? Because it is also identifying what is the electrical condition that is going on in your heart, isn't it? So again, biosensors will also be a good group of things which will give you some idea what exactly is going wrong or right into your body or any living organism, isn't it? So this is a very broad and very general kind of explanation. So one more chat, this thing was there. I could not see it. Sir, if you can repeat it, what was there? One more. Am I audible, sir, now?
Yes, sir, you are clearly audible.
Okay, what was this thing? One more, this thing was there, I
One
think.
more query is there. Simulated
As per simulated model of protein, how the protein of variable internal energy is highly unstable. Okay. Good question. So let me tell you in this way. So what happens is, according to the laws of nature, it says that if something is having inherently higher energy, it is very unstable, because it is once again go back to its ground state, and while doing so, it will lose some of its property. Okay. So in our protein simulations, we have shown you two varieties of a protein, of a same protein only. One is a mutant and another is a wild type. Now in this wild type, because we have not done anything, it is made for a normal condition, not for to preserve it for 1 year. It was made until and unless the life cycle of the bacteria is there, it will perform its duty, then eventually degrade. That was the idea of nature. Nature never wanted the protein to become shelved in somewhere in an electrode, isn't it? So in order to make it usable, you are doing the mutations. So once you once you found out the particular mutant site and you introduce the mutation, then you analyze that how the thermodynamic stability is increasing or decreasing. In the simulation, the blue color model was that we only changed a single amino acid, but the whole protein become highly stable, isn't it? Because why the protein was highly stable? Because the overall delta G of the protein has reduced to a significant extent, isn't it? And therefore it is an indication that if you do the perform same mutation in the real cytochrome oxidase protein by mutagenesis tools that we have discussed, by error prone PCR and several other tools, you can eventually find out a protein which is more stable. And more stable means what? More higher, what we call as shelf life, more higher, what we call as क्या बोलते हैं इसको, expiry date. ठीक है ना? So in this way I would like to answer it. I hope I was audible.
Yes, you are audible.
Yes, sir. Thank you.
One more question.
Achha, sir, if a protein and drug are too much stable, won't it be a problem? Okay. Once again. So too much stable is what? Because you are designing your protein, you are in control. You are in charge, isn't it? So why the simulation tools are there? So that you can predict it correctly. So once your protein is highly stable, it can also happen you have completely changed the active site dimension of the protein as well, because you are putting you are doing so much of experimental mutations you are putting, that the protein becomes so stable that it do not move. So I tell you a story. What happens is, in your human body also this kind of situation happens. For example, there is a protein which needs to be glycosylated through a particular pathway in the endoplasmic reticulum. All on a sudden, the protein got mutated and it is not getting into endoplasmic reticulum. So what will happen? It become an inclusion body. In physiology, we call it inclusion body when the protein becomes so stable that it is not able to do its usual function also. Then what will happen? Then there will be autocatalytic cleavage, and then there will be a whole lot of things which will degrade the protein eventually, and it will be of no use. Understood? So you said that what will happen if the protein or the drug, I'm not talking about the drug, I'm talking about the protein, which is unrealistically stable. So if it is unrealistically stable, it will not interact the usual way it should, and therefore it will become an inclusion body, and eventually one lysosome will come and degrade it. Understood? Or some what you call is a programmed cell death kind of thing will take place, and eventually it will get degraded. Understood? This is how I'd like to answer it. So anything else?
Okay,
It was audible, right?
no more query.
Okay.
Arundhati, you are there?
Yes, sir.
Go ahead.
It's a great pleasure for me to propose the vote of thanks for this
Oh, I
session.
see. Okay. Arundhati, I thought that you are going to have some question. Anyway. Yes, please go ahead.
Yes, sir. I thank today's eminent speaker, Dr. Surajit Debnath, for his valuable time and deep insights shared by him on protein engineering for desired therapeutic potential of peptide-based drugs. We are grateful to you, sir, for this deeply enthralling and engaging session, and I'm sure that all participants would have reaped great benefits from it. I thank all the participants for their enthusiasm and active participation. Thank you so much, everyone. See you all tomorrow in the third day of this FDP.
Thank you, coordinator sir, for giving me such an excellent opportunity to interact. There may have some disturbances in between, but my objective was to really make the session a little engaging one. Anyway, thanks a lot, everyone.
Thank you, sir. Thank you.
Okay, thank you.
Okay, sir. Okay, sir. you.